My name is Dan Malamud. I'm a professor of basic science at NYU. I have appointments at both the dental school and the medical school. And I have a research lab in New York. And I have about a dozen collaborators around the world because the kinds of things that I do actually require working with people in the areas for which the diseases we study are endemic. Historically, the very first salivary diagnostic test was developed several centuries ago by the Chinese, who were trying to decide in their courts if somebody was lying or not. And they found that if they gave somebody a dry handful of rice and they were lying, they couldn't produce enough saliva to swallow it. And it turns out we now understand why that's true, because we know what controls the secretion of saliva. But this was probably the first salivary test. The field developed largely because it was recognized that most substances that are present in blood are also present in the oral cavity someplace. And that's because the oral cavity is very highly vascularized. So the concept of salivary diagnostics really caught on when people realized that things in blood could also be measured in saliva. However, usually the level is 10 to 100 times less than it is in blood. And so that was a problem up until probably the 1970s or 80s, and that's when science gave us amplification techniques. And most people have heard of ELISAs for protein, PCR for nucleic acids. You know that if you've watched anything on TV, that you can detect DNA in saliva. You can even detect DNA on a cigarette stub or on a glass from the oral cavity. And that's because there's very little of it, but you amplify it with PCR. So using protein amplification and nucleic acid amplification, you can really increase a very small amount of a molecule to detectable levels. So we started actually looking for wound healing molecules because of the observation that both animals and people lick their wounds. And so I was looking for anti-bacterial factors, growth factors that promote healing. An interesting observation is that if you bite your cheek, it never gets infected. It bleeds, and that it heals very quickly and doesn't get infected. And that's a combination of antibacterial molecules present in saliva and wound healing molecules. And indeed, it doesn't scar either, which is quite interesting. But when the HIV epidemic began, I got interested in working on this because it seemed to be a very important problem. So we began studying HIV, and we also began modifying existing tests that people were doing on blood or urine and seeing if we could make them work on saliva. And currently, I say for the last 12 years, I've really been focusing on diagnostics for 3 specific diseases, and that's HIV, TB, and malaria. And the reason for that is those 3 diseases occur in the exact same geographic locations. And indeed, in general, if someone is HIV infected, they're going to die from either TB or malaria. So if we could diagnose those, we could treat them. It's much cheaper to treat TB and malaria than it is to treat HIV. So we have been working on rapid tests that are low cost, and the important thing for an infectious disease, I believe, is point of care. Because for some things, take diabetes, there isn't an urgency to, if you have diabetes, you don't need to do something about it that day. It can wait a week or even a month. If you have an infectious disease, you really have to treat it very quickly. So the idea of a test that could diagnose a disease at a place where you could then be referred to immediate treatment would have a big impact on epidemics of any kind, flu, HIV, you name it. If we go back 10 years, the diagnostics tests that were done in clinical laboratories had these very large instruments that were very expensive and required highly trained technicians and were very expensive to carry out. The idea of microfluidics was to scale down, and if you do something at a very small volume, everything is cheaper. The machine is cheaper, the reagents are cheaper, you need less of a sample, and it actually goes much faster. So in other words, if you're trying to amplify a molecule and you've got 10 mLs, it's going to take a while. If you have 10 microliters, it's going to go much, much faster because it heats up faster to do PCR. It's easier for the molecules to interact with each other in a small volume. So by scaling down, you save money, you save time, and what's most exciting is you increase the simplicity of the device because it's built in there in some ways. And again, there's many types of these point-of-care devices, but most of them do employ microfluidics. We could have the test up and ready to go in 2 years. That said, in order to get it approved, if we were going to do it in the US by the FDA, could take another easily 5, 6 years. So we will, because the epidemic is elsewhere, because the need is elsewhere, we will probably not start with the United States. And we are making links currently in India, in China, and in 2 countries in Africa. And we still have to get approval, but it's much easier someplace else than here.